Hey everyone, welcome to Stat's latest event, the ASCO Recap Live. I really appreciate you guys being here. I'm Adam Feuerstein, biotech reporter here at Stat. And before we get started, just a couple. Well, I don't know. Do we really need to go through the housekeeping items? You guys know Zoom probably as well as anybody. We're all PhD in Zooms these days, so we really encourage questions. So you guys know the Q&A, icon at the bottom of the screen. Please use that to post questions. Let's get lots of questions in there because we got a great panel, and I will sprinkle questions in throughout the discussion today. With that, I would like to introduce Jim Weiss. He is the CEO, founder of Real Chemistry, to kick off with some introductory remarks. Hey, Jim.
Hey, Adam. So thank you and welcome, everyone. Earlier this year, we announced that the company I founded almost 20 years ago today became Real Chemistry. Our vision is to make the world a healthier place for all, and more specifically, based on my own personal experience, a place where cancer is no longer an immediate death sentence. I have a few stats to share from this year's ASCO from our influencer tracking platform, Simpler, that we brought into the Real Chemistry family back in 2019. More than 17,000 articles were shared and nearly 10,000 individuals were involved in the digital conversations. Of this important content, there have been more than 450 million impressions on Twitter since June 3rd. The top trending term at this year's Congress was patient. We find this really notable given our focus and our mission. Real Chemistry provides clinical, corporate, and commercial solutions that meet the needs of today's healthcare stakeholders. Really, everything we do is designed to empower the patient to take control of their health and access the right treatments at the right time, equitably and cost-effectively in today's data-driven, digital-first, post-pandemic era. After today's virtual event, you will know more about the challenges and needs facing our industry, particularly when it comes to cancer and cancer care. Just look at the way we all came together this past year to defeat COVID and the amazing impact we had for people around the world. Equally impressive is what we learned from Operation Warp Speed and the game-changing mRNA vaccine platform, which we think can help program the body's cells to create proteins to fight cancer. It's this kind of coming together, even virtually, which we're all getting used to, at medical congresses like ASCO, that Real Chemistry is all about. As someone that has spent many years working with leading companies to diagnose and treat cancer, from advanced chemotherapies, antibodies, targeted therapies, combinations, and more recently, immunotherapies and cell and gene therapy, I'm more optimistic than ever that we're inching closer to adding the word cured next to more and more cancers. To that end, I'm personally committed via my work with our clients, but also with organizations like the Cancer Research Institute. Before we get into today's sessions with Adam, Dr. Rick Pazder, and three of his esteemed colleagues from the FDA's Oncology Center of Excellence, I'd love to share some personal thoughts on the current cancer landscape. We need greater awareness, accessibility, and practical scaling to make cancer treatments more cost-effective and accessible for all. Some of this was addressed during ASCO, but we can do more. We're at the beginning of the curative era of cancer as healthcare pivots its focus from sickness to wellness. We should all be excited about developments in the immunotherapy field when it comes to checkpoint or PDI inhibitors. As we know, when these checkpoints are blocked, T cells can kill cancer cells more effectively. New conditioning agents have shown promise in small studies at making cell therapy safer for patients undergoing blood cell transplantations. And this year marks the 20th anniversary, like our own, of the miracle drug Gleevec, which was developed to help the immune system target CML cells while leaving healthy cells alone. And even though Gleevec started as a leading drug for treating leukemia, we now know it can also halt GI cancers in their tracks. I'll talk more about this in a minute, but the time has come to ensure that all clinical trials for cancer and other disease states are diverse and inclusive so that we can achieve health equity now. On the heels of our second straight virtual ASCO, we learned about several new important advances in cancer research and treatment, thanks to Adam and Matt and the rest of the amazing stat staff for all the great recaps. First, there was Novartis's novel radioligand therapy that delivers a lethal dose of radiation directly to prostate cancer cells. As Adam reported on Sunday, the results of a large phase 3 study showed that this therapy significantly delayed tumor growth and reduced the risk of death by 38% among patients with advanced disease. Given the emphasis of this year's ASCO on health equity, it was encouraging to learn that the University of Pennsylvania was able to increase the percentage of Black patients seen to 16%, matching the percentage of Black people with cancer in its region. And now China is creating cancer drugs that will potentially disrupt the global competitive landscape, but still ultimately drive new and more exciting innovation in this area. Thank you again to staff for allowing me to introduce this provocative discussion between Adam and Drs. Pazder, Klutz, Singh, and Threat. They're ready to dish on some of the hottest topics in the world of oncology, including the FDA's regulatory standards and views related to new and emerging cancer drugs. We have never been in a more exciting time than we are right now. Back to you, Adam.
All right. Hey, thanks, Jim. Thanks very much for the introduction. And let's get this thing started. I am going to introduce, Rick Pazder is here. I think everyone knows him, but I'm going to let Rick introduce himself, and then we'll have the panelists introduce themselves, and we'll get right into the questions. Hey, Rick.
I'm lost here.
Nope, you're there. I see you.
I don't see you, though. Something happened here. Launch meeting. Okay, let's go here. Leave and join.
Oh, looks like we lost Rick. Harpreet, you're going to have to give Rick a lesson in Zoom.
Rick is actually really proficient in Zoom, but I'm sure he'll be with that. There he is.
Okay, I'm Rick Pazder, finally. Okay, and I'm the director of the Oncology Center of Excellence at the FDA. I'll turn it over to Harpreet.
Hi, I'm Harpreet Singh. I'm an oncologist. I direct one of our 3 solid tumor divisions within the Office of Oncologic Diseases. And I also serve as our associate director for cancer in older adults within the Oncology Center of Excellence.
I'm Paul Klutz. I'm a medical oncologist and one of the deputy directors of the Oncology Center. I oversee a couple programs, patient-focused drug development that looks at digital health and symptom and function measure, a real-world evidence program, and then I also oversee a solid tumor division and a toxicology division.
Mark.
Yeah, hi, my name is Mark Gear. I'm a medical oncologist, hematologist, also one of the deputy directors of the Oncology Center of Excellence, and also acting supervisory associate director in the Office of Oncologic Diseases.
If I may say a few words here, Adam.
You go ahead, Rick. Can I just note that I'm the only person on this event who's not a medical oncologist. So
we'll make you an honorary one, okay? For the day, okay.
Go ahead, Rick.
The reason why we invited the whole crew here is I really wanted to showcase the talent that we have in the OCE and really to showcase the people that are actually doing the work. So it's certainly not me. And there's a lot of people behind me that really do the day-to-day activities and are intimately involved in our decision-making processes. So share the wealth, folks.
Great. As I said, everyone get your questions in. I'm starting to see some fill in here, but let's start. Rick, it seems like your group is approving more cancer drugs than ever before, doing those reviews faster than you've ever done them before. And I wonder, that always brings up the criticism of whether standards have been lowered, whether you're too flexible and, marginal drugs are reaching the market. How do you respond to that?
Twofold, or manyfold, so to speak. I think sometimes people don't understand drug regulations, okay? People want to put regulations that don't exist on us. For example, we have to demonstrate that the drug is safe and effective, not that it is better than what we approved yesterday. People have to understand that we have to move on from the traditional overall survival endpoint to meet patients' needs. And that has been due to a variety of situations, smaller populations and targeted drug classes where you don't have hundreds of patients. You might have 100 patients in the United States for a particular indication. It may be because equipoise has been lost because the drug is so much better than what is out there. So I think there's a lot of misinformation that goes along. And as you pointed out, we're the most active area in the biopharmaceutical world at this time, and we're basically have a lot of activity. It's hard for most people to keep up with this, including myself, to be honest with you. And that's why we really divided most of the work up among many senior people, not just including myself. So no, we haven't lowered standards. What I think is the drugs have gotten better.
In terms of review times and the way the ability that you have to review, and maybe anyone can chime in, but it does seem like reviews, well before PDUFA dates. Do you guys have I wonder if do you have an internal clock that's different from the PDUFA date where you want a review to be done in a certain amount of time?
No, I never set a time, and neither do the division directors, that you must approve a drug by this date ahead of the PDUFA date. My point is okay, you have this work, you understand the need that's there. You should take the time that you need to review the work. I do not want anyone to say that Dr. Pazdur rushed this for some artificial time point or to make us look better. Everyone, when we approve a drug, has to sign off, and I want them to feel that they've had adequate time to look at the application, to formulate their opinions on that application. So no, we don't tell the review staff that you must approve the drug by this time. Obviously, we have meetings where we discuss things. We have a different approach in terms of reviewing these applications. And
different how? Like different from other groups within the FDA?
I think so, because we have much more of a collaborative approach where many times we bring in the sponsor even before the application is submitted or shortly thereafter, have a discussion about the application where the signatory authority is there on the application. We're discussing internally what we're going to be doing, many times even before the application is actually submitted, because we have the top line results of these applications. In other divisions, traditionally what has been done is a kind of a sequential approach. The primary reviewer reviews an application, it goes to the team leader, it goes to the division director. We don't do that. We really work more as a team. And in addition to that, we've had innovative programs, including the real-time oncology review program, an assessment aid where we have a application a documentation, I should say, the assessment aid is, where we don't ask our reviewers to do a lengthy written review. We're concentrating on really the core material, the analytical, intellectual issue, rather than them having cutting and pasting and writing 300, 400-page documents. I always say, I don't want our medical officers, our medical oncologists to be glorified secretaries here, Adam.
So let me bring, Paul, Harpreet, Mark, and you can kind of join the conversation. Like in terms of, what's your perspective from a regulator standpoint of, what cancer drug development looks like, when drug sponsors come to you with data, with meetings, with questions. What's that like from your side? Because we hear a lot from the company side, right? Companies have meetings and they talk to people and they put out press releases, but we don't really know what you guys think about it.
I would say that, we meet with companies and we look to try to help them along to create the most efficient path to get strong evidence as to whether the drug works or not work. So we don't look at it as a confrontational, we look at it as a way to try to assist in finding out whether the drug works and how safe it is. And so I've always found it more collaborative than I think maybe people might think from the outside world. That's my perspective.
Mhm.
Not to say there's not confrontation, because I think on occasion things do get contentious, and sometimes I do wonder how that's captured or reported by the companies. But to Paul's point, it is all collaborative and it is all patient-centric. And when there is, debate or dispute, it's really about us being a science-driven agency and wanting the best science and the best evidence to support novel therapies for our patients.
Without giving away too much, Harpreet, can you give an example?
Well, I can talk about, I think sometimes there are just, the agency and the company may not be on the same page when it comes to how much evidence is needed to support a particular program, or for example, a dosing strategy, or what sufficient evidence may be.
Mm-hmm.
Again, when I use the term contention, it maybe disagreement, it was a better term. And that often can and has recently resulted in some requirements or commitments from companies to, for example, explore additional doses in a post-marketing space.
Yeah.
Oh, go ahead.
Sorry, go ahead, Mark.
I was just going to add, I think the key here is the scientific, aspects of the applications, and we do see it as very collaborative because we're trying to address the underlying issues and provide advice regarding that, and that's where I think the interpretation of what is the amount of evidence or level of evidence that would support addressing a specific objective or scientific issue is what is often at hand. And I think importantly we listen to the rationale that's being provided by sponsors. So we absolutely want to hear the perspectives as to what is the rationale for an underlying approach that may be different than what someone else has used. I mean, that's where I think a lot of discussion happens, but it's also leads to advancement as well in terms of various ways to advance clinical trial designs and what have you to.
I think if I may say, you saw really a magnified version of our interaction with industry in April with these 6 ODACs, which I'm sure
we're going to get to that.
Maybe Rick can comment.
Let me,
we're going to get to that.
Let me jump in here because I think these 3 have been somewhat too politically correct for Dr. Pastor. Okay, so having seen multiple during this 20 years at the FDA, I think there's an inherent tension here. When we're giving advice to a company, we're asking what is the best program that you could bring forward? What is the most robust thing? Many times in the company's mind, they want the quickest and the least, okay? What is the fewest number of patients that you will take in a single arm study? We can't do a randomized study. Oh, yes, you can. Oh, no, we can't, okay? So, these are the back and forth that go on. And then they obviously bring in their key opinion leaders who they pay to come in to try to convince us of our misgivings, so to speak. But there's this inherent tension that always exists in these meetings.
Well, every companies put out press releases, and every meeting with the FDA is always positive, and you're always incredibly supportive of everything that they're doing. So, and then we find out that that's all a bunch of lies. So, that's why the FDA, that's why the briefing documents are so fun when we read them. Harpreet, you brought up the the maximum tolerated dose. Companies, that this sort of this trend of what's been sort of establishment is that go to the maximum tolerated dose, and recently obviously we've seen evidence of companies being asked to explore lower doses. Amgen, for instance, their recently approved KRAS inhibitor, you've asked them to look at a lower dose. Can you talk a little bit about anyone here? Can you talk a little bit about the rationale there? It seems like this is not just an Amgen thing. This is something that you're asking for all companies, all cancer drug developers. Talk a little bit about what's going on there. Yeah,
I'll talk about the Amgen drug, and then I'll turn it over to Rick to talk, and the others to talk about Project Optimus as a whole. But I first want to say this was a really landmark approval. KRAS G12C mutations are found in 13% of lung cancer patients, and this has been seen as an undruggable target for several years, despite really futile attempts. And so we were all very excited to see this data, to expedite this approval. But yes, in an effort to expedite the development program, we also felt during the course of the review that the pharmacokinetic and the target saturation data wasn't fully supportive of, we didn't feel that the company really optimally explored all the doses that they should have in an ideal fashion. And that's what triggered their post-marketing requirement to look at a lower dose to see if it would be as effective. As you mentioned, this is not just an Amgen issue. This is a cultural issue within oncology. And I'm going to turn it over to my colleagues to kind of talk about the bigger picture here.
Is when more is less and less is more. Okay. And this is the title of a paper that we're writing, and it reflects back to exactly what Harpreet was saying, is we have a culture here that more is better, and this is probably best exemplified by the fiasco that oncology underwent looking at high dose chemotherapy with autologous support that really showed no benefit, decades ago. But that was the ultimate continuation of that philosophy. But nevertheless, it still persists. What we really want companies to do, and this is like all other therapeutic areas, is start doing randomized studies looking at different doses before they go into their pivotal study. It's very, very difficult to retrofit a lower dose after the pivotal study is done for obvious reasons. You have a survival advantage, a time to event endpoint. We can't ask them to repeat a study. So we're kind of retrofitting a dose. That work should be done before the study is initiated, the registration study, albeit whether it's a single arm trial or whether it be a randomized study. And I think really one of the major things, and we realize it, and it goes back, Adam, to what you were saying, is this tension. They want to get that drug out there as quickly as possible. There's a clock on their head, so to speak. Our role is, okay, let's try to back off here. Doing a rush job here is like building a house on quicksand. And although we've been forgiving in these refractory disease settings, once you start moving that drug up into less refractory disease settings, we will not be that forgiving because you have a much different risk benefit. So it's really incumbent upon these people to figure out the dose beforehand. We're not asking anything more than any of the other therapeutic areas in the FDA.
So what would this look like kind of in a if we think about just sort of the way we sort of think about sort of the classic drug development process where, company goes in and starts doing, looking for maximum tolerated dose. They find side effects, they'll go to a certain dose level where they see unacceptable levels of toxicity, then they will back off and they'll say that's the, the recommended go forward dose. I mean, how is that different from what you would like to see companies develop the drugs?
I don't want to consume all the oxygen during this conversation, so others please jump in, but obviously they could do randomized studies looking at a variety of doses because they're seeing responses usually at a variety of doses. And one of the problems that we have, small numbers of patients are entered at those dose levels, okay, or varying doses, maybe 10, 15 at that, okay, at best. So there's overlapping confidence intervals, but there's definitely an escalation of toxicity. What needs to be done is a randomized study looking at these various doses, looking at response rates to determine which one would be the one that they want to carry forward. Even with single arm trials, if that is their preferred method of getting their drug approved on a single arm trial, they could do a randomized study early on looking at 2 or 3 doses. So at an interim analysis, select a dose, okay, and then continue that onward in a single arm study, utilizing those patients and counting those patients that were entered already at that dose level. So you have a continuum going on. So here again, we're trying to work with sponsors. We're aware of the need of patients for these therapies. But here again, unless we address this issue, we're going to get into a very difficult situation of not knowing what the correct dose is, subjecting people to unnecessary toxicity with very little benefit. I don't know.
Is this a requirement? Is this a requirement or a recommendation, a suggestion? Like what from the regulatory side? I mean,
We are going to start making it more of a requirement to look at this quite closely, okay? It's in their best interest. It is in their best interest to do this.
Absolutely. And I would just add, I think, here in terms of being able to do randomized trials at different dose studies or at different dose levels or cohorts at different doses with more patients available, that opens up opportunities, to really select a dose that is also supported by additional pharmacokinetic information, pharmacodynamic, modeling target saturations. I think Harpreet brought up this point as well. And that is something that could aid in dose selection. And then there's also the potential opportunities down the line that may not be realized at the beginning of a dose finding study, but with more full characterization at different doses, you have the opportunities for potentially expanding opportunities for additional dosage regimens, some that may be more convenient, for example, or as we found during the COVID-19 pandemic, actually could be related to safety issues. So, for example, extended dosing of monoclonal antibodies and decreasing inpatient visits, while in other times might be for convenience, but it could actually be additional safety measures for a patient as well. And that more full characterization early on, can allow some of these other opportunities, in later development.
Yeah. Adam, I think, I was a phase 1 investigator. I published the first phase 1 study on Taxotere, for example, from MD Anderson. We have much more information available to us now on these newer classes of drugs. It's not just the clinical appreciation of toxicity. We have the ability to look at dose saturation or saturation of targets, pharmacokinetics more extensively. One of the other things that we're emphasizing also, and it goes back to Jim's comments on wider representation of ethnic and minority groups and the aged and other underrepresented groups, is to start including them on these trials to look very early at, are there differences here? And I think that's one of the other important points that needs to be brought out in this whole issue of dosing. Because if we do see very early on in dosing, there may be ethnic, racial differences, gender differences, age differences, then we need to start paying closer attention to that and their representation in the actual registration trial.
So I'm going to go to a question. We had a question about someone wanted to know, Rick, whether you're going to keep your promise from the 2019 AACR meeting to approve a Chinese developed checkpoint inhibitor.
I was misquoted on that.
Okay, so you can set the record straight.
I am setting the record straight. Okay, somehow, and this is like urban legend that got out there by one reporter publishing something. We'll go nameless, not from STAT. Okay.
Thank you.
Okay. I was just talking about the reality of the world, and what happened was that it came out that I was endorsing this or I was enthusiastic. I was just saying, this is what's going to happen, okay? These companies are going to come in with very similar PD1-like drugs or PD-L1 look-alike, so to speak, and they're going to go after the same indications because we already know what the effect rate is. These studies can't be done in the West because these drugs are approved, so they're going to do them in China. Risk is minimized because they already know they work, and they also know what the effect size is. So I never said that I was going to approve a drug, nor was I enthusiastic about it. All I was just basically doing is saying, folks, this is the reality of the world. Wake up and smell the coffee here.
But since that time, and I know you can't talk about drugs that are under review, but there is a PD1 checkpoint inhibitor that you are reviewing right now, developed in China. There will probably be another one submitted to you soon. So how do you, and these are, again, to be clear, these are drugs that were developed and tested in China. So they don't have U.S. patients. They weren't tested in the
And we
in
have
Western
approved
in the
other
West.
drugs.
How do you
Approved
look at
other
those data
drugs.
differently? How do you do that?
Well, there's ICH guidance on this, okay? And one has to take a look at intrinsic and extrinsic ethnic differences. Intrinsic is the population similar biologically. Extrinsic would be is the care that was delivered to these patients, similar to what was existed in the United States. Let me remind you, Adam, that most of our data that is coming from approvals or that's submitted in registration trials is occurring outside of the United States, okay? Yes, there is US component to that, but the majority of that data is coming from outside of the United States, almost in most of the major pharmaceutical companies' dossiers for drug approval. So this is not a unique situation where we are only seeing studies done in the United States with only US populations. That rarely exists at the present time. So those are the issues that we have to face here. Is the population and the disease similar and representative of the United States, okay? And is the biology in that group of patients that were studied abroad the same? So we're talking about intrinsic and extrinsic ethnic differences, so to speak. I can't go into anything more on that. I don't know if Harpreet wants to jump in the lake here, so to speak, in the pool. She handles lung cancer application, so that's coming to her desk.
Yeah, those are coming to us, but I think I think when we talk about these Chinese studies, in my mind, they're not all created equally, and I say that because some of them, Adam, you kind of mentioned this idea that there's this kind of recapitulation of existing data, and that is happening. But we're also seeing benefit in terms of different tumor types like nasopharyngeal cancer, in which these trials could not be enrolled in a US or even European or even a mixed population. And again, because we are a global, we are an international country, and we have many Chinese Americans, it's not quite appropriate to say a Chinese-based study is not reflective of the US population. Perhaps better terminology would be it's not broadly reflective. But, so I think you can't view all of this data through the same lens, at least from an oncologist standpoint or an unmet need standpoint or even a clinical practice standpoint. Now, how that translates into regulatory action, I think, remains to be seen.
Well, along those lines, as a follow-up, I mean, how do you look at the comparability of these drugs, and whether they're all the same? There's differences. Are the differences in the drugs? There are differences in the studies. If there was a Chinese-developed PD1 or PD-L1 that wanted to get into lung cancer in the U.S., how would they do that? And would they have to do another study? Like, how do you guys think about that?
Well, as I said, we have taken data from other countries. Does it represent, from a biological point of view, the same activity as one would expect in the United States? And that you could take a look at, obviously, the clinical information that comes from the study, as well as PK issues, target saturation, et cetera. So even beyond, a response rate or a survival curve or time to progression endpoint, that is interesting. What is fascinating about this is doing a head-to-head comparison of these drugs, which I don't think will ever be done, but that would be an extremely interesting study to be done. And this is one of the unfortunate situations that we have, and I've been railing on this for the past 7 to 8 years, so to speak, of the proliferation of these PD1 drugs. And despite my admonitions, there's not less, there's more, and it's blossoming here, so to speak. And we have to ask ourselves the question is, do we need all of these drugs that they're kind of competing against each other rather than advancing the field?
Well, it seems now the big trend, and we saw some of this at ASCO. We're seeing it more at the medical meetings, not just this ASCO, but ESMO and all the meetings. We're seeing a lot more adjuvant data on adjuvant use of not only checkpoint inhibitors and immunotherapies, but targeted therapies. In the adjuvant setting, in the neoadjuvant setting, and it seems like there's this debate between, disease-free survival and survival. Can maybe a regulatory perspective from you guys on that? on kind of the strength of data and sort of any challenges to develop these drugs kind of in the earlier lines of therapy?
I'm going to let Mark and Paul address that because I've been talking too much and they've just been quiet.
I mean,
You guys it's like being called on by your teacher.
Yeah, it's fun. I would say, disease-free survival is, and even metastasis-free survival, some of the more novel endpoints where the disease is going to take a very long time. There's few events. The trials will be very, very large. Are reasonable endpoints. They signal a complete change in the natural history of the disease and what the patient's going to be experiencing. So disease-free survival has been an endpoint we've used across tumor types. And so I think it's not a novel endpoint to us. Mm-hmm. And so I'm sure that will continue to be used.
That note has passed, Adam. Okay, it passed decades ago when we started using it in breast cancer and in colon cancer. So for us to say, well, we can't use it in lung cancer, and I realize that some in the lung cancer community wanted overall survival, that would be holding lung cancer up to a standard that we didn't held any other disease up to, and one would have to ask themselves the question, why? Considering it is a lethal disease when it's metastatic.
I don't know if you ever go on Twitter during medical meetings and watch lung cancer oncologists argue about DFS versus OS. It gets pretty heated sometimes. It's like a cage match sometimes on there as people start arguing about whether there's enough data to support the use of these drugs. It's kind of fun. Just on accelerated rules, and I think Harpreet, you mentioned this. The recent ODAC meeting that y'all had, the 3 days of. I mean, that was a marathon. But what came out of that? Like, what are the lessons or that came out of those 3 days?
I think that's a Rick question, but I'll say that my division was not. We luckily were not part of this marathon because we had a sprint at the beginning, so to speak, in which we were able to, in consultation with 2 companies, have them voluntarily withdraw their indications in small cell lung cancer. And frankly, I think the lesson that I took from the ODACs was that that was probably the appropriate choice, because in small cell, the landscape had dramatically changed and immunotherapy has moved to a frontline setting with a survival advantage.
So I'm not going to let you escape. I'm not going to let you escape about now. Was this a thing where you were told them we are withdrawing this drug, or was this a consultation?
Not at all. We don't have the authority to do that. I think that's something that I think we'd all like. This experience has probably taught us that we'd all like to change, but it absolutely was probably a series of conversations with both companies. The alternative was to go to an advisory committee meeting to ODAC, and I can say that.
I'll quote Peggy Hamburg in a phrase that she taught me: "For those that do not see the light, they will soon feel the heat."
Yes. So they got out the kitchen.
I mean, most of the accelerated approval withdrawals, almost all of them, except for one, were voluntary withdrawals by the company. So I think, what we should take away from this ODAC is that there wasn't that many new things. We've looked at accelerated approvals now several times, once in 2011 at an ODAC, again in a paper that we did, 25 years of accelerated approvals, looking at endpoints, et cetera, and then again in this ODAC. And I think, it shows that we're monitoring the program. We don't want drugs to be on the market for a long period of time until their post-marketing trials are done, and we can understand whether the benefit's been verified. And I think one of the take-home messages we've all been talking about is the strength of an accelerated approval application. One of the variables we really look at is whether that post-marketing trial is well underway, because it is very hard to accrue to those trials, and that's what takes a long time. Even in 2011, when we looked at the 5 outliers that had been on the market for over 8 years, nearly all of them hadn't had their post-marketing trial underway, and some of them hadn't even had the protocol completely done. So it was something that we learned over time, and I think we're doing better with.
But yet there were 6. I'm sorry, go ahead, Rick.
Question for you. June, what were the dates? At the end of June, it's going to be the 10-year anniversary of guess what? the Avastin hearing in breast cancer.
Yes,
Okay.
well, that's the precedent that everyone brings up, right?
And I want to answer your question here of why we brought these, and a lot of it was transparency. Okay, people have got to understand the PD1 drugs, albeit my criticism of them and the duplication of trials, etc. I have to really emphasize the benefit that the American public has seen with these drugs, okay? And no one will deny this. This has transformed the field of oncology in almost every single disease. But nevertheless, the accelerated approval were done. Some of these trials failed or did not meet their primary endpoints, as we like to say. And we wanted to have transparency to let people know what we did. This area of work has been since the last 6 or 7 years since the first PD1 drug was developed. There was over 76 or something like that indications that were approved. This is the most active area in the pharmaceutical industry's history of the PD1 drugs, bar none. Okay, there is no other therapeutic class, and this has all developed over a short period of time. Now, one of the things that we have to realize is we're going to be on a learning curve here. And the FDA could be part of that learning curve and avail these important drugs to patients, or we could say, no, you can't have it until we get, these studies that have survival endpoints or randomized studies. So we wanted to really show that there were some people that won their primary endpoints, others did not. What was very interesting though, because we had this proliferation of studies, okay, some in the exact same indication, we saw some of the drugs win, some of the drugs did not win, okay? And here again, there's never been a head-to-head comparison of these 6 or 7 approved PD-1 drugs. But nevertheless, we have this body of data, and most people think that these drugs are very, 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 very similar, okay? And we were able to analyze this data, and there were many problems and differences in trial design. Some trials were underpowered, and that's why they failed. Some had the wrong primary endpoint. They should have chosen overall survival rather than PFS. They had the wrong hierarchical testing. They should have looked at a PD1 positive population than an ITT population. So there were many reasons. I want to emphasize to people.
Yeah, but don't you think that critics will say, it's up to the companies to design, in consultation with you, to design the right clinical trials, and the stakes are high here. And if those studies don't work, then why don't you just remove those drugs from the market?
Because our obligation is to the patient. There is a patient here also that needs these drugs, okay? And therefore, we want to make sure and work with the companies. A failed trial does not mean a failed drug, okay? And this is something that people have got to understand. And people simply can't get it through their head that you could have a very active drug, but if you make a mistake in designing your trial, you may not be able to demonstrate that. And this is particularly important in this PD1, PD-L1 arena, where we have an evolving field of knowledge. With cytotoxic drugs, we had a 50-year period of time here to figure out what was going on. This is an intense period of rapid drug development, and we wanted to really make sure that it worked well. And I think despite all of the warts that have occurred in this field, the American public has gotten great benefits in almost every single oncological disease, especially those that had few and far therapeutic options available to them.
Yeah, I would just add that, now there's, I think 21 or over 20 different diseases for which there is a indication for an anti-PD1, PD-L1. And, that's over, 7 now different, PD1, PD-L1 targeted antibodies. And this also includes 2 tissue agnostic indications. So I think, Rick, you made the point that there have been benefits, of such an active area of development based upon not only the science, which led to the initial, but also, the innovation that happened within oncology in general, and with the various therapeutic classes. But the one other point I just wanted to make, on this topic is, a failed trial doesn't necessarily mean a failed drug, but also a withdrawal doesn't necessarily mean a failed program either. So, from an accelerated approval perspective, there is an expectation that some of these trials that are ongoing will not confirm benefit here. And that's just part of the program. When you're thinking about what is the evidence, required to support, support an accelerated approval, it's all substantial evidence. But, when thinking about the effect sizes on endpoints, for example, in later, confirmatory trials on other endpoints, there's going to be some that just do not, confirm in the trial, clinical benefit. So, that's unfortunate, but, unfortunate expectation. But those programs are for, there for the patients. And, overall with, the amount of activity, we've seen great advances.
So sticking with accelerated approval, but switching diseases, you may have heard that your friends over in the neuroscience side of the FDA approved this Alzheimer's drug on Monday.
How do you spell it?
Did you hear anything about that?
How do you spell it?
It's called Aduhelm. Yeah, just a little bit of news. So, obviously that was obviously big news, but also big news was the framework in which the decision was made. It was granted accelerated approval. And I hear, Rick, that you were sort of brought in. What role, if any, did you play in this whole thing? Yeah.
I don't know. Well, I listened and I was asked to comment on this. And here again, I want to make it explicitly clear. I am not a neurologist. I am not a neuroscientist. I don't know anything about tau. I don't know anything about amyloid. Okay, so I want to make that perfectly clear. But if you believe, okay, that this amyloid reduction is a potential marker or a surrogate, if you want to use that word, which I hate, okay, or an earlier endpoint for eventual clinical benefit, then accelerated approval would be appropriate, okay? End of discussion. We have approved drugs in oncology on single arm trials. This fact that there was a positive and a negative trial, well, that's one of the reasons that we have accelerated approval. And I think I probably have the most experience of probably anyone in the world with this accelerated approval process or conditional approval, if you want to take a look at the European programs. And I have a more expansive viewpoint on it. I look at it as a way to modulate risk, basically. Okay. And here again, the substantial evidence in oncology, for example, when we approve a drug on a single arm trial, is the fact that, these tumors would not shrink by themselves. So there has to be some biological activity there that the drug is causing an effect here. And most oncologists would believe that if you have a reduction of a certain size and a certain duration, that would be a potential benefit to patients. I don't know if that exists in the field of neurology. I leave that up to my neurological colleagues and the people that made that decision. But from a regulatory perspective, if one has the belief that a reduction in amyloid is a potential surrogate for it, this is an appropriate pathway to approve the drug and modulate the risk. Now, would we have liked to have a clinical trial that was up and running? Yep. Okay, that's sure. I'm sure everybody would agree to that. But we've been in other similar situations in oncology where we've accepted this.
I guess the situation there is where it's hard for people to sort of wrap their heads around, and where the criticism is coming is that they did run two big studies, right? Outcome studies, and one positive, one negative, and the positive one was whatever you want to say about that one, and now we're sort of going. It's almost like we're going backwards. Instead of,
we have taken a look at studies, for example, in oncology, where we're not impressed with their primary endpoint and approved it on a single arm, just taking a look at the single arm experience of one arm of the randomized study. Here again, Adam, I can't go into, because I didn't review the entire application. I'm just giving my viewpoint on a topic that I discussed with the review team and with the others in CEDAR.
So a question again from the audience. Someone wants to know about the disclosure of complete response letters. In those rare instances when you guys actually reject a cancer drug, I know it doesn't happen very often. Seems like you prove them all. But in those cases when you do reject one, obviously you issue a complete response letter. You know, companies tend not to disclose the full contents of those letters. We sort of get a kind of cleaned up summary of maybe of what you want from a company. And there's a lot of spin in those in that disclosure. What do you guys think about that? Would you be in favor of, and I know a lot of people are in favor of more open disclosure, maybe disclosing complete response letters. Any thoughts on that?
I definitely would. Harpreet?
All right, so Rick Passer is on record. He wants complete response letters disclosed. I like that.
Oh, you better believe it. There's no reason that American patients put their lives on the line here to go on those studies. People from other countries did this. It should be very well known. We owe it to the patients to make that disclosure. Harpreet, do you want to comment on that?
Agree. No, I mean, I agree. I mean, it also sometimes it's not. It may, you know, I've issued a few CR letters myself, and it's not always about the clinical data. Sometimes it's about manufacturing, and I think for that not to be disclosed, you know, can perhaps lead to negative perceptions about the drug development program and the clinical data, which I think, you know, could be mitigated by public disclosure of those letters.
We save a lot of ODACs.
What'd you say, Paul?
I mean, I think that part of the transparency that we're able to do is if we have a challenging application, we take it to an ODAC.
Right.
A lot of work. And it would be nice, honestly, if we could just show, we've already written an ad nauseum as to why we felt that it wasn't appropriate at the time for approval for various reasons, as Harpreet says, but you know, we have a lack of transparency, and therefore oftentimes we have to bring it to advisory committee meetings. Not all the time is an ODAC related to a problematic application, but it is one of the ways we can provide transparency.
Right. Like, I don't know how often you guys keep track of like what companies say publicly about stuff. Because I think one of the things, you know, Paul, speaking of ODACs, I mean, I think one of the sort of revelatory aspects of an ODAC meeting is when we do get to read the briefing documents, and then we see the timeline of the interactions that the company had with you. And we see in there where there's disagreements, or where you say, you know, we told you to do X and you did Y. And, you know, and of course we were told, like we meaning the public were told, oh, no, the FDA totally wanted us to do Y. You know, that stuff gets people pretty angry and upset. And do you feel like, I mean, I don't know if there's anything that you can do, but I wonder if you guys like track that, like if you see that and there's nothing that you can say about it, but you know that it's happening.
I think one of Rick's lasting legacies will be how he opened up the FDA oncology group to, you know, all sorts of external engagement. So we end up doing that a lot in external engagement. We talk about stuff, and sponsors will say, or patients will say, or anyone will say, well, we heard FDA requires this. And we say, that is actually not true. And so one of the ways we can do this is through external engagement. And we probably do it more than any other therapeutic area, I would say, at the FDA. So that's one comment.
And that's about product type or class, you know, just general principle, not product specific, obviously, but yeah.
So someone wants to know, you know, given the high cost of cancer drugs and sometimes limited effect, should quality be taken into consideration in the review process?
I favor keeping our determination of safety and efficacy separate from any cost considerations. That's a whole different issue. I think we have enough problems of determining safety and efficacy without the determination of cost, so to speak. So that's for other people, because cost could change on a dime. Somebody could decide, wake up the next day and cut the price in half, or quadruple it the next day, as we've seen examples of that, obviously, from people in the pharma world. So to speak.
And there's been a few questions about diversity and access. You know, it's been touched on here. I mean, are you satisfied with the current state of trial enrollment? Do you think companies should be doing a better job of diversifying patient enrollment? And then, you know, also on the access side, you know, I mean, and is there anything that the FDA can do about that other than to just recommend?
Well, we have, I'll ask Harpreet to talk about this. We have a whole program on, she runs the part that has to do with elderly, but there's a whole part of project equity that we have looking at how we're going and aiming sponsors to really enroll and have enrollment plans for ethnic and racial minorities. But Harpreet, this is probably your field that you would want to talk on.
Yeah, Adam, this is really important to us even before, this year's ASCO theme on equity and the FDA as a whole and also OCE. But yeah, we have 3 projects: Projects Community, which is run by Ria Blakey; Project Equity, which is run by Dr. Lila Pashona J; and then Project Silver, which I run. But really the overarching theme here is that, what can we do from a regulatory perspective to promote and just make it easier for all patients, and not just race, age, and ethnicity, but also things like geography and sexual orientation to have access to clinical trials. And we are proactively asking them when they first come in to discuss their plans for clinical trial design. And just to pick up on something Rick said before, it's not just about the pivotal trial, it's about the early phase trials, the dose finding trials, making sure that different race, ethnicity, and age categories are included. So I think we are seeing some movement. We saw some recent publications from industry actually that they're listening and paying attention. And I think one very important thing that we're doing is asking for post-marketing data. We've done this several times in the last year or so when a clinical trial data set doesn't really reflect the population that cancer drug is meant to serve. And I think you're going to see us use this tool more frequently.
And we're asking people in their meetings to really have a plan, of how they're going to address this. And stay tuned. At ODAC meetings, we will be asking for this information to be presented both in the briefing documents as well as in their presentations also. We're very serious about this. This is not just lip service for this year and then we're going to forget about it. One of the problems that we have, obviously, I can only speak for oncology. I'm not sure what goes on in other therapeutic areas, but as I mentioned before, a lot of the enrollment, 70%, 80% sometimes are coming from outside of the United States, from Eastern Europe, from former Soviet republics, Western Europe, etc. It doesn't, the remaining people that are in that trial are sometimes 20%, and we have decreasingly small numbers of ethnic and racial minority groups. And that's why we really have to work on this issue, because as we have a more international perspective, we have even a greater difficulty in analyzing adequate numbers of patients.
So we have just a few minutes left. So Rick, let me ask you a question. How long are you going to keep doing this? What's Rick Pazer's next few years look like?
Every day is a great day when you're my age, okay? And I thank God every morning that I wake up, okay? And I just take day by day, so to speak. When I originally came to the FDA, I arrived in 1999, and I met with a group. We had, and Bob Temple was the office director at that time, who had been at the FDA for 20 plus years. And I said, I don't plan on staying here for more than 3 years. Okay, so I was wrong. Okay, and I really enjoy the people that I work with. And at this point in my career, I'm not financially motivated, obviously, and I take great pleasure in the people that I work with and their careers. Having been in medical education for a long period of my life before I came here, and having many of my former fellows and trainees take leadership roles, I enjoy seeing my children, so to speak, at the FDA, and all of these guys on this screen here could be my children by age, so to speak. I take great pleasure in their accomplishments.
Well, with that, I think we'll wrap it up. Paul, Harpreet, Mark, Dr. Pazer, Rick Pazer, thank you so much for your time. It was great. It was a really great discussion, and thanks for joining us today.
Thank you.
Thanks, Adam.
Thank
Thanks so much for having us.
So I want to thank our sponsor, Real Chemistry, and thank Jim Weiss for the introductory remarks. And let you guys know that our next STAT event, I think it's a related to the big bio convention. It's on Thursday, June 17th. So please tune into that. And with that, we'll say goodbye. Thanks a lot, guys.
Bye-bye.